Hello, everyone. I'm Dr. Neil Druve, Health Program Specialist of the NIH CounterACT Program, and I'm joined today by Dr. Chardell Spriggs to present this webinar on PAR 23027. It's a funding opportunity to support basic research on chemical threats that affect the nervous system. First, I'd like to go over a couple of housekeeping items. We are recording this webinar, and afterwards, we'll share the recording and the slides with all the registrants. At the end, we will take questions that are submitted in advance through the registration or any new questions that you might have. You can enter your questions into the chat at any time. Specific questions about your proposed research project may be done on a one-on-one basis with NIH staff at a later time. So, if you have specific questions about your research, feel free to email us. We have our contact information in this slide presentation. With that, I'll turn this over to Dr. Chardell Spriggs, Program Manager of the NIH CounterACT Program.
Thanks, Neil. Welcome everyone to the webinar. I'm Chardell, a Program Director in the Division of Translational Research at NINDS, and I also manage the NIH Countermeasures Against Chemical Threats program. Today, we will cover a few things, including a brief description of the NIH CounterACT program, this new R01 CounterACT funding opportunity for basic research, and then we'll provide some tips that we've seen help people have successful applications. The CounterACT program has funded research since 2006 with the mission to understand fundamental mechanisms of toxicity caused by chemical threat agents, and then the application of this knowledge to develop promising therapeutics for reducing mortality and morbidity that is caused by these agents. And we do this with a focus on exposures that occur in civilian mass casualty situations. We're the only program here at NIH with this focus. This means we often have many people that need to be treated at once. Our therapeutic approaches cannot include pretreatments, and they need to be fast-acting and easy to administer by first responders that may be encumbered if they're wearing personal protective equipment. We also use models that test efficacy that mimic these real-world conditions. CounterACT has the overall goals of improving the nation's medical response capabilities, supporting cutting-edge research to improve the knowledge base, and we develop optimized lead compounds for transition to advanced development. So exposures to chemical threats are a real-world problem. Chemical threats are toxic compounds that could be intentionally used, such as in a terrorist attack, by governments for chemical warfare, or recreational drug adulteration, such as when rodifocone was added to synthetic marijuana. But there are also accidental exposures related to industrial production, storage, or shipping. And the Ohio train derailment is a recent example of that. As you can imagine, with so many possibilities of threat agents, CounterACT is a trans-NIH effort. Grants are managed across NIH at these ICs based on subject matter expertise, but we also work collaboratively to leverage the experience available at other NIH institutes as needed. CounterACT funds research on over 200 chemicals of interest as identified by the Department of Homeland Security. We group these chemicals into toxidromes, such that the chemicals within a toxidrome have similar modes of action and toxic effects. One benefit of this approach is that a single medical countermeasure with broad spectrum activity may prove effective against multiple different chemical threats within that same toxidrome. And we've listed them here for you with a few examples of each. This funding opportunity focuses on those toxidromes that have either primary effects on the nervous system, and that includes the cholinergic warfare toxidrome, the pesticides, and the convulsants. We're also including toxidromes such as the anticoagulants, blood agents, that also have secondary toxic effects on the nervous system. And the relevant toxidromes are here in red. And actually, we'll have to add one more correction because this should also include the hemolytic and metabolic toxidromes as well. So the CounterACT program has a suite of funding opportunities that span the research that we cover. Our space starts in basic fundamental research. These are funded through R01 and R34 mechanisms. ICs that oversee the toxidromes for these have published several funding opportunities to cover those. You can see that here listed in the bottom left of the screen. We also have R21s for exploratory developmental research. And then we have a UG3, UH3 phased cooperative agreement mechanism. This is our highest level of funding. In the UG3 phase, you narrow your therapeutics down to a lead compound, and the UH3 phase is used to optimize that. Today, the focus of this webinar is on the CounterACT R01 basic funding announcement that focuses on chemical threats that have primary and secondary effects on the nervous system. And that's PAR 23027. And so this funding opportunity targets the first part of our mission, and that is to understand fundamental mechanisms of toxicity that are caused by chemical threat agents. So when an acute chemical threat exposure occurs, there are a number of mechanisms involved that lead to pathology, toxicity, and poor outcomes. And these include persistent long-term effects. This is magnified in our situation when we are usually dealing with delayed treatments from exposure, such that even when the standard of care is delivered, and these are only in some instances where there is a standard of care, we still see this pathology that leads to long-term effects. This funding opportunity covers a spectrum of interest, and Neil is going to really get into this in the next couple slides. But we want to make it really clear that we're looking at short-term toxicity that happens after acute chemical threat exposure, including all the mechanisms that lead to pathology, all the way through long-term effects. And at this time, I'd like to turn it back over to Neil.
Okay, thank you, Chardell, for the overview of the program and for describing the unmet need that this funding opportunity is meant to address. And I'd like to dig a little deeper into that. So this funding opportunity, PAR 23027, is titled CounterACT Basic Research on Chemical Threats that Affect the Nervous System. And the goal of it is to support projects that elucidate the mechanisms of toxicity of these chemical threats, new manifestations of toxic exposures, and potential targets for therapeutic development. The scope of the supported proposals will be basic research that is relevant to understand the mechanisms of toxicity and pathology of these chemical warfare agents, toxic industrial chemicals, and pesticides that have primary or secondary effects on the nervous system. And this includes long-term effects from acute exposures, as Chardell just mentioned. Now, if you have questions about which COC toxidromes are covered, I'm going to refer you back to the earlier slide in the presentation, the ones that Chardell highlighted. And that's key for this funding opportunity. Those are the COCs with primary and or secondary effects on the nervous system that will be considered responsive. Basic research on the mechanistic basis of other COC toxidromes are covered in the other funding opportunities that are mentioned on that slide. So here we've listed examples of research that would be within scope of this funding opportunity. And this could be research to identify the molecular mechanisms of acute toxicity to chemical agents and identification of relevant biological markers and targets for therapeutic development. And also to elucidate the mechanistic basis of long-term effects from acute exposures to chemical agents. You could propose to develop in silico, in vitro, and in vivo models to elucidate known and unknown mechanistic pathways of toxicity. And you can use those in vivo models for natural history studies of toxicity of chemical agents. Another example is to identify the mechanistic linkage between signaling molecules and their biological targets, and to decipher their functional relationships and physiological roles to allow better understanding of toxicity of acute exposures. You could research the mechanisms of toxicity that are not addressed by the current standard of care when the current standard of care exists, the mechanisms of toxicity that are not addressed by that standard of care. And that includes mechanisms or targets that improve refractory status epilepticus, post-SE neuropathology after acute exposure to chemical threats. And you could expose the mechanisms of toxicity that cause neurodegeneration or alter neurodevelopment and affect the brain development or the blood-brain barrier, neurogenetics, neuroexcitability, or neural circuitry. And please keep in mind all of these things. These are all examples. You're certainly not limited to just what is on this slide. This is to give you a flavor of the kind of research that will be supported. And lastly, I would like to mention that we do encourage applications that propose research on more than one chemical threat within a toxidrome. Next slide. So I've just told you a bunch of examples of the kind of research this funding opportunity supports. And here are some specifics about that support. Applicants can request up to $300,000 per year in direct costs, and the project period may not exceed 3 years. This is an R01, so the entry criteria are for rigorous preliminary data that supports the hypothesis. In this case, for the mechanistic pathways you are proposing to study, and that there is a relevance to the chemical toxicity. So this funding opportunity has 2 more receipt dates. The next one coming up is May 30th, and then after that, it is October 17th, both from this year. Now, I do want to emphasize examples of research that are non-responsive to this funding opportunity. And that is, at least one of the chemical threats you propose to study must be on the current DHS list of chemicals of concern. Otherwise, the research you're proposing will be non-responsive. Also, research proposing to study COCs with primary effects on the pulmonary, skin, or eye systems, or to study opioid-based chemical threats are considered non-responsive to this funding opportunity. If that is what you're studying, you should apply in response to the other basic research funding opportunities listed earlier. Chronic chemical exposure models are considered non-responsive. This funding opportunity only supports research on the health effects after a single acute exposure event. Additionally, clinical trials are non-responsive. And finally, and this is important, studies on the creation and validation of candidate therapeutics are non-responsive. Early stage translational efforts for therapeutics development are supported by the R21 and the UG3 UH3 funding mechanisms in the CounterACT program. But this funding opportunity is for basic research. And I should say that, the applications that are non-responsive will not be reviewed. So now I'll turn this back over to Dr. Spriggs to finish up the webinar.
Thanks, Neil. We're going to ask that you please read the specific notices listed here on enhancing and implementing rigor and transparency in NIH-funded research. Pay close attention to describing scientific premise, which is now the rigor of the prior research, specifically the quality of that research, including strengths and weaknesses of data you include in your application. Make sure you present a rigorous experimental design, including proper statistical analysis and control of bias. Address sex as a biological variable. If you cannot use both sexes or you see no need to, have a good explanation why not. And too expensive is usually not an acceptable reason. Address authentication of key agents used in your research. This includes the chemical threat agent. Saying you purchased something from Sigma or other supplier is not sufficient. You will need to run an analysis to verify the identity and purity of key compounds. The emphasis here is on key agent and does not include minor reagents and buffers. Please do not use the vertebrate animal section for experimental details to circumvent page limits, intentionally or otherwise. Okay, the reviewers are not required to consider this section for that purpose. Not only will this result potentially in less enthusiastic review scores, but the review officers may also pull your application and not allow it to go to review if you stuff this section. Include all letters of support for collaborations. If you're working with someone and they're a key person on this grant, please include that letter from another institution. Please include a letter from your biosafety committee saying that it's okay to do this research at the institute. And make sure you address biohazards and facilities that are needed for research on restricted chemicals such as chemical warfare agents. And this should include a plan for containment and disposal of biohazards. So a couple of further application considerations, and this is just from us seeing, what works and doesn't work, what sometimes gets flagged, and it's just not enough preliminary data to establish scientific feasibility. Okay, make sure your preliminary data and the literature supports your hypothesis and the proposed studies. Make sure you are presenting sufficient details so that the reviewers can evaluate the approach that you are proposing. And once you're done writing everything up, make sure that the aims and the work that you propose to do actually meets the goals of the project. And if needed, be sure to address interdependence of aims. And that's usually a bigger problem for translational research, but if you have that here too, just make sure you talk about it a little bit. Part of that can be just discussing alternative approaches to your research strategies that are presented. If this is an MPI application, make sure you describe, especially then how the team will communicate. And then also, we're always interested in having investigators come from other research areas, investigators that are new to the field of chemical countermeasures research. And we also like to support early stage investigators and new investigators. But this is an R01 mechanism. So you do have to consider what being successful in this award will do to your ESI status. And just make sure you weigh that against your other options. Lastly, we want to leave you with scientific contacts. If you have any questions about research that you want to propose, we are more than happy to make ourselves available and can set up Zoom meetings to talk to you one-on-one to make sure you have a responsive application. Neil mentioned some other funding opportunities the program has. So if you're in a different Toxidrome and you want to talk to one of them, you may find their contacts on our website. And the very last thing we want to point out is the need for diversity in science. So if you currently have a grant from NIH and you have 1 to 2 years left, please look into getting a diversity supplement to your current grant. Okay, and this is a very robust program at NINDS in particular, but it is NIH-wide. And I think at that time, we can just address any questions you guys have for us.
Okay. Thank you very much. So I encourage everyone, feel free to put your questions into the chat and we will address them as they come in. I will read the question and then Chardell can provide an answer. And we can go from there. I will let you know, we have a couple of questions, but we don't have many right now. So, once we're out of questions, the webinar will be over. So please go ahead and write your questions now. So first, we'll start with a question that came in earlier. Chardell, how can someone find out if their proposed chemical threat is allowed for the purposes of this funding opportunity?
Sure, thanks, Neil. That's a very important question, and it applies to all CounterACT funding opportunities, not just this one. What we ask you to do is to contact us. And it's on this screen as well. I understand this question came in earlier, but you are strongly encouraged to contact us because we may only fund research that is on that chemicals of concern list. There are some versions of it published elsewhere. We are not allowed to publicly disseminate that, but very happy to talk to you. You may even email us if you want. Maybe you don't have time for a call and you just want a quick answer. Just shoot us an email and we'll let you know.
Okay, that's great. So moving on. The next question is, how many applications can someone submit? Can they send in multiple applications as long as they're studying different chemical agents or potentially for different mechanisms? How would that work?
So the rules for this funding opportunity are the same rules for NIH. Okay, so you're not limited to the number of applications, but you may not have 2 applications in that are not scientifically distinct and under review anywhere. The only other thing of importance is that, CounterACT has a limited budget, and we, do consider multiple factors, including portfolio diversity, when we are determining our pay plan. So, happy to see the applications come in. This funding opportunity specifically says we're also welcoming you to include more than one chemical threat in your proposal, because that may actually review better. Reviewers may see it as more bang for the buck, but no promises. So no limit as long as they're scientifically distinct. But please make sure you're putting your best applications forward. It's definitely quality over quantity.
Okay, thank you. Another question is if, for the kind of research that is described as being covered within this funding opportunity, could someone apply to the standard parent announcement with that same, I guess maybe the question is not, can they apply to the standard parent announcement for this research, but is there a reason why they should apply to this funding opportunity announcement instead of to the parent announcement?
Okay, that's a good question. So the parent does not pull from this set of money. So CounterACT is funded through a congressional appropriation. It's a set-aside bucket of money specifically for funding research to advance knowledge on chemical threats and exposure to chemical threats. I think the CounterACT program did start with going to regular review panels, and that didn't turn out so well. So our funding opportunities are PARs, and that R means that we have specific review requirements. And the SROs for these convene special SEPs or special emphasis panels. We have to bring people in for every review cycle that have specialized expertise in these areas to make sure we get a good review. It's a lot of work, and we, love our review officers for doing it. So while you are more than welcome to submit any research to the parent, it won't have these same limits. It won't have these same review criteria, and it pulls from a completely different bucket of money.
Okay, that's great. All right, so great. Everyone's asking their questions. Keep them coming in. One that came in is I'll just read this. Is the use of standard of care treatment allowed when using OP-SE models, organophosphate status epilepticus models, or does this announcement support pure OP toxicity research in the absence of any supportive treatments?
Right, this is an excellent question. I'm so glad you asked this question. And you may use the current standard of care, or you may exclude the current standard of care. And it's up to you to make the case why or why not. But it is allowed. If you want to use the current standard of care, like maybe you want to see what's left. What else do you need to target that's not being targeted by the standard of care? Or maybe you think the standard of care is hiding something, that you need to not include it to research another mechanism of toxicity. So just make the best case you can. And the short answer is you can have it or you cannot.
Okay, that's great. So next there's a question. I guess I'll ask this question next, is if you can clarify what is expected for authentication of key biological or chemical agents, especially if it is a commercially procured agent.
Sure. Another good question. Key agents would be, your DFP or your antibodies, things like that. We've had people order things from Sigma. Okay, so there's a reviewer who no one reviews, but he loves to use the example of how he ordered one thing from Sigma, spent 6 months not being able to get it to work, and when he finally ran a mass spec on it, it was a completely different compound. So those are the types of things that we would like to see you do. Something as simple as running a mass spec. If it's going to be, a key reagent or biological in your research to avoid those types of mishaps. So I hope that answers the question.
Okay, yeah, that's great. Next, there's a couple of questions about I guess these are more about other kinds of or I guess chemical threats that are in other toxidromes. So one is that pulmonary agents are listed, or we listed them in the covered agents, but how would someone work with that for lung injury if at the same time we're saying it's non-responsive for this funding opportunity?
That's a great question. So those agents are not non-responsive to the CounterACT program. And we have funding opportunities in place to specifically serve the need for the pulmonary toxidrome. And we will be happy to direct you to those. So if you're working with something that falls under the pulmonary or the opioid, dermal, any other toxidrome other than those that have primary and secondary effects on the nervous system, there are special R01s and R34s on the street to cover all of the toxidromes for the CounterACT program. So it is non-responsive for this, but we do have a funding opportunity for you. So you can shoot us a quick email, and I think Neil is dropping them in the chat right now, where you can link out to those. They're going to have different criteria, but they're for a similar budget and length of funding.
Yeah, that's great. And there, the links are also going to be in the slides that we mail out to everyone. So you'll have these links. Okay, the next question is is also related to one of the other funding opportunities. This one's for the opioid chemical threat one. The question is that funding opportunity states that 6 projects will be funded, and does that refer to per grant cycle or per year?
Yeah, that's a good question. I would say reach out to Kiran, and we can, his email address is on our website. And as the subject matter expert for the opioid portfolio, he would have to answer that specific question about a funding opportunity that's not this one. So happy for the question, and we can connect you. If you want to send me an email with that question, I can make the introduction if that's easier, or you can find his name on our website. But very happy to steer you in the right direction.
Okay, then now I'll just tell you, this is the last question that we have that anyone submitted. So, if you're hoping that someone else is going to ask your question for you, the answer is they're not going to. It's your time to ask the question. Okay, so this last question is, for this funding opportunity, can someone evaluate a known mechanism for SE to determine if that same mechanism is relevant for OP-induced SE?
So yes, that's a good question, and we have not placed limits on the mechanisms that you can interrogate. You just have to include that OP. And so to find similarities in known mechanisms that are involved in is perfectly acceptable, but I would also advise you to just reach out to us, since this is a specific science question, and we can have an exchange of emails or a short call to discuss a little further.
Okay, that's great. That brings us to the end of the questions, and so that brings us to the end of the webinar. Thank you very much, Chardell.
Thank you, Neil.
I will, afterwards, when we have the video available, I'll mail the slides and a link to the video to everyone who registered. I want to thank you for your interest. And, again, if you have questions or you want to follow up, definitely feel free to email us. Have a good day.
Thanks, everyone.